I'm ChulHee Choi, the CEO of ILIAS Biologics. I founded ILIAS Biologics as a professor at KAIST, Korea Advanced Institute of Technology and Science, and developed ILIAS proprietary technology for exosome engineering, so-called Explore, in my lab. I could have chosen to license this novel technology out to a third party. Still, I knew there were countless hurdles along the way to be commercialized, and most of the companies quickly gave up in the middle. The exosome itself was in the early phase, so it couldn't seem easy to stick to the end of development through the long and rough journey if the IP holder did not have scientific knowledge and passion. I started my career as a neurologist in a hospital where I could gain hands-on experience translating drug delivery systems into clinical levels. I did not want this promising technology to become rusty, so I had adequate experiences, knowledge, and sincere attachment to the technology. Therefore, I decided to leap by building my own company. This is how I got here.
Excellent. So, as we know, exosomes transfer biological messages from donor cells to recipient cells, and that makes exosome a therapeutic platform that delivers therapeutic materials to the target tissue or cell. So, your current research mainly focused on this hot topic. So, could you please share the role of exosomes as novel therapeutics for biology, technologies, and clinical applications aspects?
Yeah, that's a wonderful question. So protein drugs that we can load into the exosomes vary regardless of their sizes, including enzymes, antibodies, and signal transducing related proteins. As such, exosomes can load desired proteins, which is a technology can be extended active pharmaceutical ingredient, so-called API. Most pharmaceuticals are antibody-based protein drugs, but proteins cannot penetrate cell membrane, so they are limited in developing new drugs. On the other hand, the exosomes can solve the problem of intracellular protein delivery by loading therapeutic proteins inside the phospholipid bilayer. In particular, they can freely pass through the in vivo barriers such as blood-brain barriers and amnion, and thus have potential as a targeted therapeutic. In addition, exosomes can overcome the limitations of protein delivery using nanoparticles and have the potential as a therapeutic agent for various diseases with minimal toxicity, a high encapsulation efficiency, drug stability, and a simple manufacturing process.
So, can you please tell us about your current research focus at ILIAS?
Yeah, at ILIAS, we dedicate ourselves to researching and developing therapeutic exosomes, particularly engineered exosomes for better clinical efficacy. While most companies have been trying to use naive exosomes itself, derived from stem cells as therapeutics, a few companies like ILIAS have been trying to upgrade this efficacy by focusing on three critical points. The first, improve therapeutic efficacy. The second, optimizing drug delivery technology. And lastly, overcoming the limitations of current production technology of naive exosomes for more scalable and stable production, so-called manufacturing. So these are three most essential factors in developing clinically and commercially viable therapeutic exosomes. Therefore, we are focusing on answering the three questions: what therapeutic substances to load into exosomes, where to target and deliver the therapeutic exosome, and how to enable scalable and stable production.
Then it seems that it's very important to look at more detail the hurdles and importance of these three focus areas of your research. Can you tell us about that one by one?
Yeah, I want to tell a little bit about the treatment efficacy and loading part first. So we may not achieve therapeutic efficacy by simply loading cargo into exosome. It is essential to focus on effectively delivering the active proteins to target cells. The space for drug loading is physically limited in exosome, so encapsulated molecules are not large, especially for protein cargos. So although we may load limited numbers of cargos inside an exosome, we may improve the therapeutic effect by increasing the target set delivery rate. So as a result, it is essential to consider how efficiently to deliver the cargo into the target cells and maintain its function. So we need to be aware that it may take some time to get well-designed protein cargo. So for the question for the second part of question about the targeting and delivery. So for targeted delivery, the exosomes have natural tropisms specific to certain tissues and cells depending on their mother cell line. We can utilize these characteristics for targeted delivery. On the other hand, it can also mean that we may not deliver to a specific target of our interest if exosomes are not naturally distributed in that tissue. To overcome this problem and increase the versatility of our platform, we are developing an exosome surface engineering technology, so-called ExoTarget, for targeting moiety displaying. At the same time, we are expanding our cell library, allowing us to target a wide range of tissues and cells. The last question about scalable production. The exosomes are a very stable drug delivery tool because they maintain their physical structure under various conditions and keep protein cargoes safely in the bloodstream. However, for actual commercialization, scalable production and quality control steps are crucial. We successfully established industry-level production of GMP-grade exosomes with acceptable QC attributes ready for preclinical and clinical trials. That is called a pure exo technology.
Now get to the what would it be if you point out one hurdle to overcome leading this research and development in this novel innovative area?
So one significant challenges we face in the early stage of translating initial research findings into clinically viable development is the validation of POC, proof of concept. We must make sure to prove the reproducibility of our result in any setting over and over. Since the engineered exosome is a relatively new area, many researchers and companies meet the deadlock due to failure of proving the reproducibility of their initial research result. It is one of the most common and severe challenges for researchers to move novel therapeutic candidates to step into the commercialization stage.
So, since you've been working on this project for so long, if you try to summarize the significance of your current research, how could you make it as a one answer?
As a one answer. So the academia and the industry are already aware of the huge potential of exosomes as an emerging technology and their marketability. What matters now is how to improve the three focus areas above to successfully translate the current research advances into therapeutics in clinical use. Improving current issues in the loading, targeting, and production of therapeutic exosomes is critical. Considering research findings in preclinical stages of engineered exosomes and clinical results of naive exosomes so far, we expect no major safety problems in its clinical use. Therefore, once we solve the issues in the three factors above, the novel exosome therapeutics will move into clinical use and can be used to treat patients with speed. The significance of our research focus is this. It is a crucial stage for us to translate the basic scientific knowledge accumulated in the last 10 years into clinically viable treatments.
So, looking forward to the future development, you share your insights about how this industry or research to evolve.
So all novel modalities accompany concerns and expectations throughout the journey to explore their way to become clinically viable options. However, despite the current hurdles, we can expect explosive advancement of engineered exosome treatment if we find the solutions. Most importantly, we can develop therapeutics that can reach the undruggable targets inside cells. Therefore, I think exosomes may open a new era for targeted delivery, like precision medicine. So, although no exosome drugs are currently available, the development speed is getting faster as clinical trials with exosomes have skyrocketed. Therefore, in the coming years, we expect many exosome drugs to be approved and become critical technology in the drug delivery area.
Thank you so much for your insight. Now, would you please tell a little more about your company, ILIAS Biologics?
ILIAS Biologics is a biotech company dedicated to develop a new treatment modality, engineered exosome therapeutics, founded in 2015 as a startup inside KAIST, a Korea Advanced Institute of Technology and Science, where I have been a professor. ILIAS has successfully created 3 proprietary platform technologies: Explore, ExoTarget, and Pure Exo. The Explore makes it possible to load large therapeutic molecules into exosomes, in which can subsequently target previously undruggable intracellular pathways. The ExoTarget allows active targeting of its engineered exosomes to specific organs of cells. And lastly, Pure Exo enables the manufacturing of high-purity exosomes on a commercial scale. The ILIAS accelerates innovation through 2 tracks. The first, in-house pipeline development in inflammation and CNS area. And second, research collaboration with industry partners interested in leveraging ILIAS's unique platform technology. We have 78 employees as of November 2021. About 80% of employees are working on R&D. ILIAS Research Center consists of 3 central departments of early discovery, translational research, and manufacturing. We've successfully completed our Series A and B funding last November and now preparing for Series C funding. We plan to initiate our global clinical trial of the first pipeline drug, ILB-202, the anti-inflammatory treatment, in early 2022 and list the company for initial public opening in Kosdaq in late 2022.
Then finally, the last question. Could you please share your advice or message with our researchers and companies who want to enter into this new field?
I think exosomes have substantial potential thanks to their limitless expandability as novel treatments and game-changing drug delivery technology. Almost the infinite expandability of the pipeline using exosomes is a huge opportunity and a great challenge that cannot be fully explored nor overcome by any single company or just a group of researchers. Therefore, open innovation is the key to actualizing and accelerating this paradigm-shifting new modality for patients in the end. We at ILIAS are waiting for partners in academia and industry to lead the change together. Come and join us in the journey to paradigm-shifting medical advancement.